because she's kind of like an academic family member. We go way back to, we started grad school about the same time. And about the same time. We're all at Duke University.
Perfect.
And Anne has always been on an amazing trajectory of interdisciplinary research. She basically had like 3 advisors during her time at Duke: Jim Beaver, Jan Santanovic, and Ritas Vogeles, the latter who which was a mycologist, or is a mycologist. And so she had this very independent track from that time. She worked on the ecology and genetics of arbuscular mycorrhizae, produced this really cool thesis. But so she had these multiple tracks, and it wasn't clear exactly whether she would go more plant or more fungal. But fortunately for the mycological community, she then got a Miller Fellowship to go work at Berkeley with John Taylor, who was a mycologist. And so she's been on the mycological track for a long time. She's now the president-elect of the Mycological Society of America. She was on the faculty at Harvard for 9 years, and since 2015, she is a professor of botany and bacteriology at the University of Madison, Wisconsin. And since 2017, she's the Vilas Distinguished Achievement Professor, which is quite an achievement. And throughout her whole career, she's been a real role model to me for interdisciplinary and broad research. And so she has multiple research tracks, including the spore dispersal and the biomechanics of that, which she won't talk about probably at all today, but she has a really amazing paper called The Captured Launch of the Ballistospore, where she demonstrated that the spores of mushrooms are shot off at 10,000 Gs. This required about 100,000 frames per minute photography. No, that's frames per second. Big difference. Yep. And so I could go on, but I should probably just hand it over to Anne, who's going to talk today about the biogeography of body size using population genetics to delineate fungal individuals.
Thank you, Tim. Thanks a lot. Thanks for having me. It's a wide room, so I'm still going to try to look at everyone and march around, I guess. I don't know if that'll keep you awake or be really distracting, but you can let me know afterwards. And I'm going to start the talk. So this is actually a talk that involves data that spans about, almost 15 years now. I haven't published this data before, but you're going to see some data that was collected actually in around 2004 or 2005, and I hope it'll make sense by the end of the talk why I didn't publish it at the time. And so I wanted to start the talk by acknowledging the people who were with me at the beginning of this adventure. And those people are Franck Richard and Ben Wolf. And I don't know if you can see, but they're holding this gnome or elf creature that one of our collaborators gave us. It's actually dyed with mushroom dyes. It's one of my most prized possessions. And this is Hugh Cross, the technician. And this is my daughter, who's now 15 years old. So you can see, what it was all like back in the day. Oh, I wonder how far I can go. So one of the issues with giving a new talk is that I haven't quite gelled it into discrete sections to give it titles. So I'm going to try to talk about what the discrete sections are and make it make sense as I go along. And so I want to set up the story initially by doing 2 things. One, talking about the biodiversity of fungi. And second, teaching you about the system that I've been working with for a long time, which is the death cap, Amanita phalloides, which is a mushroom, a fungus that's invasive in California. But to start the story, I want to tell you, if you don't know, that we know extremely little about the biodiversity of fungi. So I'm talking with someone who's writing a book about how many species there are on Earth, and she said to me, that's the thing that I can't figure out is how many species of fungi there are, because she said every piece of literature I read seems to tell me a different number. And we think there are something that we have names maybe for 74,000 species, but we think there are something between 1.5 million and 10 million species of fungi on the planet. So you can see there's just a kind of absurd discrepancy between what we have names for versus what we think is actually out there. And of course, this knowledge is being spurred not only by careful work on the part of taxonomists, but by the revolution that's been enabled by molecular technologies, by the PCR machine and the advent of meta-barcoding and metagenomics. So the fact that I can stand here and give you a range that encompasses different orders of magnitude should tell you really the state of our ignorance about how many fungi there are on the planet. So we live in a changing world. We know that. I don't think there's any point in pretending that somehow the biology we do right now is the biology that we might have done 100 years ago. And so one concern that we have as biologists is what's threatened? What kind of biodiversity may be disappearing? I find that to be a hard question when you don't even know what biodiversity you have at the moment or 100 years ago. Sometimes people say we need more baseline data. I think we shouldn't fool ourselves. The time for baseline data is gone. It's lost. Whatever we're collecting now is baseline at this moment, but it's not the baseline data we would have collected 100 years ago, and it's not the baseline data that we would collect 100 years from now. Nonetheless, if you start looking at statistics, and these statistics are taken from an older paper in natureserve.org, you can see, you can calculate, and I did these by hand, so I'm not completely wedded to the exact number here. But just as a tool, a teaching tool, something like 15% of the plants in the United States are described as imperiled. So if you use the same sources of data and you try to understand how many species of fungi in the United States are imperiled, you get a number that's 0.4%. So either fungi are less likely to be imperiled than plants, or once again, and this is what I would argue is the truth, we know so little about fungal natural histories that we don't recognize an imperiled species when we see it. And the truth is that quite a lot of things may have gone extinct under our feet, and we don't know. So when we think about biodiversity in a changing planet, invasive species are part of the conversation. And here you're looking at two rangers who are ripping out garlic mustard, and I'm guessing for many people in the room, garlic mustard is a familiar invasive species. So what I'm trying to tell you is that actually when you think about invasive species, you may think about invasive fungal pathogens that destroy elm or destroy chestnut, but you probably don't think about invasive fungi that are not pathogens. And that's been a great part of my work over the last 15 years, is working with this species of fungus, which is invasive on the west coast of North America. You're looking at a poster that warns you that picking and eating wild mushrooms can kill you. These images are of Amanita phalloides, the death cap. And this is the species. This poster is put up in California at various parks to warn people as they enter the parks if they're trying to find porcini or chanterelles or something, that Amanita phalloides is there. And every year, regularly, a few people eat this mushroom and die. Most recently, 2 years ago, when a whole family actually ate phalloides. And the family recovered, but the 2-year-old toddler did not. So, and part of my message here is that this is, to my knowledge, one of only 2, actually, species of ectomycorrhizal fungi, so it's a symbiont of plants, but really only one of 2 non-pathogenic fungi where we understand very well that it's an invasion, and it's invading, otherwise unperturbed forests in California, and I'll show you that. But I don't think that that's indicative. I don't think that this is a rare story. I think it's just a story that we happen to have paid attention to. And it's anyway, this is a large part of my story today. So why is it so, why don't we know about more invasive fungi? Why isn't this something that we could go to NatureServe and just pull up a lot more data on? And that has also been a part of my work through the years. And the story with Amanita phalloides, and I suspect the story for a lot of fungi that might have been picked up and carried around the world, is that our understanding of the species of fungi and species concepts has changed a lot through the years and shifted in complicated ways that makes it complicated for biologists to do the accounting, basically. So here's an image of the European death cap. Reverend Barclay is actually how you say his name, published a book called British Fungology in 1860, and this is a very useful book for me because it shows you the typical color of an Amanita phalloides. It has this kind of olivey brown color. This is the European species of Amanita phalloides, the death cap. So American mycology really began only in 1902. Is the first serious publication of American mycology. It's McIlvaine and McAdams' 1001 American Fungi. And what you're looking at is an image from this book. These are all the deadly Amanitas sitting with the skull and the elf with his toxicology textbook. And this is Amanita phalloides, according to McIlvaine and McAdam. And I think you can see right away that this has nothing to do with this. This is also supposed to be phalloides. And this is really the crux of the problem. So actually, if you go to the herbarium off campus and you look for specimens of Amanita phalloides, you will find them, even though I can tell you that Amanita phalloides does not grow in Michigan. But if you go to the herbarium and you look, you'll see several boxes full of specimens that are labeled Amanita phalloides and collected in Michigan. So this is another guy, Murrell, who also, this is according to him, Amanita phalloides. As you can see, it bears no resemblance at all to the European phalloides. And this quote from 1934, Jacob Lang, accurately summarizes, I think, to a certain extent, where we still are. When an American mycologist who knows the European species from descriptions or books only, he's never traveled to Europe, it's an era before travel was so common, faces the problem whether a particular fungus is the one named Agaricus whatever by Fries, he will often be at his wit's end and may count on his buttons to settle the case. So the message that I'm giving you is one of confusion. We have a confused record of our local biodiversity, and that confused record is still very much with us today. And so, for example, you can see this collection made in California near San Jose University, I think is what that is supposed to be, collected in 1912. And it says, this is an old name for Amanita, Venenarius phalloides. So this person is claiming in California to collect a death cap, just like your local herbarium, but the color is all wrong. The cap is supposed to be cream color with brownish center. So again, a message of confusion. I've actually sequenced this specimen and confirmed that in fact it's not an Amanita phalloides. Nonetheless, by 1973 on the East Coast and 1977 in California, people who did know the European phalloides because for whatever reason they had been to Europe, had carefully recorded that that European fungus was in North America. So certainly by the 1970s, it was with us. And since then, I've done a lot of work, and I'm going to just summarize it for you briefly in the next set of slides to catch you up to date on the biology of the death cap as we understand it now, and the true death cap, the European Amanita phalloides. It's been introduced from Europe to North America, to South America, Australia, New Zealand, Africa. So it's now found on all continents. If you do a model, this is one of these MaxScent models where you take abiotic variables from the sites where you see the death cap growing and you project that across a map to understand what suitable habitat might be. You see in colors that are lighter, including parts of Mexico, which apparently are very good habitat for phalloides, though we haven't seen it there. But you can see what are supposed to be the good habitats, and then red marks a known Amanita phalloides population. So you can see its distribution more or less up and down the West Coast. Here is the center of invasiveness, and on the East Coast as well. It has very different ecologies on the East and West Coast. So Ben wandered through a forest. This is a native quercus forest on Point Reyes in California, and every time he saw a mushroom, he clicked his GPS. So you can see that it's really common and abundant and widespread in this habitat. This could have been done any day, for example, in December. On the East Coast, this is the New Jersey Pine Barrens, and this is a patch of forest that was planted by the Civilian Conservation Corps probably in the 1930s. And you can see that on the East Coast, this fungus is not invasive. It's there, but somehow it's constrained in this really interesting way. So you only ever see it within this patch of planted forest. It's sort of a fascinating dynamic that we don't understand at all. So it has different ecologies in the different places. The California habitats where it's invading, it's not co-invading with an invasive plant. That's not what's going on. It's shifted its host, so it's now growing with a native oak endemic to California, Quercus agrifolia, in habitats that don't appear to have a lot of other disturbance going on. So this is just an image to show you what a typical habitat looks like in California where you see the death cap. And where you see the death cap, you see a lot of the death cap, not just 1 or 2 mushrooms. So here we went to 2 forests, and in the course of a short period of time during the season where you would see a lot of, by the way, I use the word sporocarp interchangeably with mushroom. Sporocarp is the more formal name for a mushroom. Carp means body, it just means spore-bearing body. I don't like the word fruit body because fungi don't make fruits, so sporocarp or mushroom. So anyway, you can see that in both of these sites, if you look at the biomass of the Phalloides mushrooms, they dominate. So above ground, when you're walking around in these habitats, when Phalloides is fruiting, there are a lot, dozens, 30, 100, hundreds. It's hard to describe how, and they're sort of disgusting, so they're deadly. And just it's a really interesting experience to walk in an invaded habitat. And there are very few other fungi. Below ground, not quite the same story, but I'm not going to talk about it today. The sporocarps, the mushrooms themselves, are much larger. So here's the average size in California of a mushroom. Here's average size in Europe, where it's native, on the East Coast, where it's found but is non-invasive, and in the Pacific Northwest. So they're really big. And you can find them at any time of year. Oh, so one mushroom can produce over 80 billion spores in a day. And you can find them at any time of year. So here in Europe, fruiting is really located in the end of summer, fall, more end summer in the north, in Sweden, for example. And then by the time you reach Corsica or Sardinia, you might reach November, December. But in California, any time of year. And this is a little bit, there's something hidden in these data, which is that in the forests where you find Phalloides, actually you really find them in November, or December, or January in California. But Quercus agrifolia, the coast live oak, is often found in people's gardens, or their backyards, or the daycare center, my daughter's daycare center. So when they water, then you find the death cap emerging as well. So these records, like in the middle of the summer, are coming from well-watered, like Coachland Hall. For those of you who've been at the University of California, Berkeley, there was always regularly a few Phalloides that popped out on the trees near Coachland Hall. So no, the fruiting isn't really restricted to a particular time of year. And this is just again to emphasize that the death cap is not unique. Even though people haven't really, and still haven't really talked about this issue of invasive mushrooms, if you will. When we went to the literature, my colleague Elza Belinga, who speaks 6 different languages, so she could go to a lot of what we would call the gray literature, like the Amateur Mycology Club of Florence. And she could say, okay, what have they found? If you read in Italian, and they'll say, oh, I found this, and I've never seen it before. In 100 years, we've never seen it, but now we see it. And so we say, okay, that's probably a record of a new thing that's growing there. And if you look in the literature that way, for many countries where there's yellow, there's data. And so, for example, for Brazil, you can see there's something between 64 and 120 introductions. And we made this database public, so if you want it, you can go to the publication and download it and see what has been reported as introduced or invasive for your favorite country. Introductions, invasions are concentrated in the southern hemisphere because pines are not native to the southern hemisphere. And when foresters started planting pines for timber to make pulp or toilet paper or whatever, their trees did not grow until they would ask a forester friend to go collect soil at Blenheim Woods outside of Oxford. And that forester there would send a tin of soil from Oxford to South Africa. They would put it around the pines that they were growing, and the pines would grow. It was actually some of the earliest evidence that these were beneficial relationships. But in the process, a lot of fungi got moved around a lot of places, and the stories get just as complicated as you might imagine. In Colombia, for example, there's a fungus in Amanita muscaria where the seeds came from Mexico, and the plant is Mexican, but the seeds came from South Africa, and the fungus is European, but probably also came from South Africa. So that's a background, and that's what I've been working on for a long time, is simply trying to carefully document the natural history of an invasive fungus and ask a lot about its dynamics, what it's doing. So it feels a little bit like, I don't know what it feels like, but it's, if you work with plants and you work with invasive species, I think some of what I'm telling you must be very familiar in terms of the behavior, but just know that we haven't had it before. We haven't had this information for an invasive fungus before. And so today what I really want to talk to you about with an old data set and a new data set is something that also seems like one of the first questions you should be able to ask and answer about any invasive species, which is how is it getting around, right? How does it reach new habitats? And very specifically, I've been thinking for a long time that it must use the massive number of spores that it creates to reach new habitats. And we have an idea in our head that spores are small and that they should travel very long distances. And I think for a lot of us, that's probably still the intuition. And part of that is right, and part of that is not right. So spores are small, they should be able to travel. And then I'm also really interested in this question of how big an individual is. So think of a mushroom, but of course the body of the fungus is in the soil. It's called a mycelium, it looks like a network of threads. So that body underground is also, you know, in some ways the main part of the organism, right? What's going on with that mycelium below ground? So those are the questions I had when I set out actually in 2004, 2005 to map individual mushrooms in Europe, in California, and on the East Coast. And Ben and Frank helped a lot. And then use genetic fingerprints. And at the time, I was using a very cutting-edge technique called AFLP. And for people in this audience who are graduate students who've never heard of that technique before, just know that it's a genetic fingerprinting technique. That's really all we need to know about that for the moment. And I was going to use this genetic fingerprinting to distinguish one individual from another individual. And it looks like this graphically. So you go to a habitat, and you get XY locations for all the mushrooms in that habitat, and then you do the genetic fingerprinting. And there are kind of two possibilities. One possibility is that every mushroom appears to be more or less its own genetic individual or genet, and then you would say, oh, wow, there are many small genets, small individuals below ground, and sexual dispersal via basidiospores or spores. Or you could find that actually there were very few genets, and in this case you would say it's growing underground. There's a large body underground spreading with its mycelia, and then you see lots of mushrooms popping up from that individual body. And in the literature, so genet mapping is not as common now. Back in its heyday, there was a lot of this kind of work, and basically you could find many different kinds of strategies. So what did I find? Well, first, let me tell you that I sampled in California, the East Coast, France, and Serbia. Those are the places that I went. And, let me try that again. It doesn't like my picture of Serbia. Or I can give my other talk. That must be my computer, I think. That's not you guys. Okay. So there are 7 populations, and I put populations in quotation marks. I actually teach a class called Foundations of Evolution, and we spend a lot of time talking about the word population, so I'm always a little hesitant to use the word, but what I mean by that is just spatially distinct groups of mushrooms, but sometimes the space is 90 meters between populations, sometimes it's a kilometer. So 7 populations in California, 1 tree in France, where my colleague Monique Gard collected 72 mushrooms. New Jersey, this same site that you saw before, and various places in Serbia. So I'm going to go right to the data. These are the two this is Point Reyes National Seashore. This is Tomales Bay State Park. Here's the San Andreas Fault. Here's San Francisco. Berkeley's over here, just to get you situated. And these are 4 populations at Point Reyes National Seashore and 3 at Tomales Bay State Park. And we did all of those. I'm not going to show you all the data, but I want to show you a bit of the data. So using AFLP, this genetic fingerprinting technique, we discovered that this pattern was really one of lots of relatively small genets. So here's one snapshot of the data, but actually this is the more common pattern, where every mushroom is its own distinct genetic individual. So picking lots of mushrooms is like picking lots of people. Every person's different, every mushroom is different. And we found that robustly almost everywhere that we looked. Here's one, and I'm changing all the axes on you, and I apologize for that. But otherwise you can't see the data. So we do find some mushrooms that belong to the same genet, but in general, the dominant pattern is of many small genets. And so when we do the comparative biogeography, when we ask, so there are two implications for this, by the way. One is that the mycelia below ground are not large. The other is that it's sexual basidiospores that are establishing new individuals. And that's the only way you can kind of get that pattern. So if we go to other places, so here's the New Jersey population in that planted forest that was planted by the Civilian Conservation Corps. If you look at these mushrooms, you get the same pattern. And I'm not, just for the interest of time, I'm not going to show you all the data. But basically, we find the same pattern wherever we go. So here's one image of Serbia. And this spans quite a long distance. So there are actually 10 mushrooms here and 4 genets. And then there's 2 mushrooms very close together here. So the dominant pattern is one of small genets, small individuals. And the thing that's really interesting to me about this, actually, I have no idea what time it is. Okay, I guess I don't, whatever. I'll either end, oh, the clock is over there. I see it, okay. I don't want to run late. So one of the other things we learned along the way doing this, by the way, and just as more or less a random point of interest, is that these spores are slightly unusual in that they have multiple nuclei per spore. And so here you're looking at a spore, and can you see the 2 little eyes looking at you? And we don't know what that is. It doesn't have anything particularly unusual about its genetics, we don't think. So we think it's just 2 mitotically derived nuclei per spore. But the Amanita are the group that seems more like, we know the most about it, so I don't know if it's more likely or what's going on, but it seems to be an invasive species quite often. And this feature is a feature of the genus. So I just wonder if there's a connection between these spores having 2 powerhouses inside them, if you will, that makes it more likely somehow to successfully establish from spores. That's a wild speculation. But I just find it so interesting that it has multiple nuclei per spore. So now we get so when I was thinking about publishing those data, a strong criticism I got from Monique Gard, my colleague, was that by only taking one snapshot in time, I had no idea how to contextualize the data. Maybe if I waited, the vegetative mycelia below ground would grow and they would become much larger. So maybe I was capturing a young population that would look different later. And I thought that was a fair criticism, and I didn't know what to do about it. And so I basically just sat on the data and did nothing about it because I couldn't figure out what to do about it. And then in a long, twisted, stochastic series of events, this kind of manna from heaven money landed in my lap. And I decided that what I should do with it is go back to the same habitats, exactly the same places where I'd collected before, collect mushrooms, and this time, because we're no longer in the land of AFLP, collect genomes and try to understand genet size and structure 10 years after I had originally done it, because 10 years is a long time, and if the pattern has changed, it's enough time to see the pattern change. So in 2015, we had a big expedition, and now you're looking at the more modern crew. Here's Jacob and Zach and Holly and Kat. And we went out and went to the same places and mapped and collected mushrooms and sequenced their genomes. Not as many samples as we could do with the AFLP because genomes are still slightly expensive and actually difficult to manage bioinformatically somewhat, but nonetheless, a pretty good sample size. And so we sampled the same populations in California. We also took some of the mushrooms that we had done AFLP on. I had carefully archived the tissues, and they were lyophilized, so they were in really good condition. So we took genomes from exactly the same samples. So sometimes, I'm not really going to talk about it, from the same sample we have AFLP and genome data, which if anyone wanted to write a methods paper on that, it would be a really good data set for that. So, and we sampled actually in 2014 and in 2015 then from the same populations in California. And then also now, so we were very opportunistic about how we sample in Europe, because in Europe it's rare, actually. It's not so common to find phalloides. And at this point in time, we had some populations in Portugal. So now I'm going to tell you about some populations in Portugal. So here in Europe, here are 3 populations in Portugal. And these actually mark singletons, a single mushroom that was collected, and actually often these are archives coming from Kew Botanical Gardens. And so there are a range of ages, but anyway, we just chucked them on the sequencing run, and I'm not going to talk about them too much, but it's also becoming a rich resource for understanding population structure of phalloides in Europe. But these are our genomes from Europe, and then our genomes in California are really concentrated at the sites that I told you about already. And then this one sample, which is one of the earliest samples of Amanita phalloides in Point Reyes National Seashore that was collected in the 80s, I think. So we wanted to see, actually, I don't think I'm right on the date, but we wanted to see how this historical specimen fit in. Okay, so for the genome jockeys in the audience, 86 mushrooms with Illumina, HiSeq 2500, insert sizes, paired end reads, 60x coverage, which is deep. So we have really good data, really, really good data, and that became important. So really deep sequencing. We did a couple of PacBio. Our reference genome is a Portuguese mushroom, which we did with PacBio and Illumina. We filtered, so then we did variant calling. So we basically called the SNPs, the single nucleotide polymorphisms, among all the specimens. Again, we're trying to get a genetic fingerprint just with more up-to-date methodologies. And so here's some of the data on the raw variants that we had, and we filtered that to 288,000 high-confidence SNPs. And so that's really the data set that we're working with. So what do we find? I'm just going to, again, just show you some, not all of the data in the interest of time. So here's the same population that you looked at before. This is 2004. So actually, you've seen these data before, but when you saw the data before, they were with AFLP. Now you're looking at the same data with SNPs, with genomes. And you can see that every single mushroom resolves into its own individual. And we get, unsurprisingly, higher resolution with our genomes than we got with AFLP. AFLP didn't have enough information in it. And so this is what it looks like in 2014. We don't have these sites, unfortunately, mapped in exactly the same way. So I don't want you to get the impression that this is an invasion front. It's not. We don't understand exactly the relationship between these 2 maps, but I was there both times, so I know that we're in roughly the same area that, as you know from the beginning of the talk, is heavily invaded by phalloides everywhere you turn. And this is what the data looked like in 2015. So there's no sense that in this habitat where phalloides is very frequent, that it's getting larger through time. And in fact, I think that the way to think about the death cap is that it's more or less an annual weed. In mycology, we do not use the words annual and perennial. But I think in this case, that's what's going on. I think every year, these mushrooms produce copious numbers of spores, and every year, they grow a mycelium. It would be very interesting to understand the relationship between that mycelium and the tree with which it's associated, but we don't understand that. But every year, then, it makes a sporocarp, and it sheds its spores, and the cycle starts over again. It turned out that, and let me go into the SNP data, just because this is also an important issue. So one of the concerns that you might have is that somehow we're overcalling SNPs, and so we're getting, you can go in too far in either direction, right? You can throw away too much data, you can keep too much data. So to just talk about that a little bit, these are the AFLP data. I'm not going to focus on those. These are the SNP data. These are Euclidean genetic distances among the mushrooms in any given population. So here you're looking at California. Here you're looking at Portugal. And so if you are looking down at this end, two mushrooms are more similar to each other. On this end, they're less similar to each other. So the two most closely related mushrooms in the SNP data set are only 90% similar, with 25,000 SNPs differing between them. So for two things, in other words, the two mushrooms we have that are the most closely related differ by 25,000 SNPs. So they're really different from each other. So if we talk about throwing away data, so these were when you're playing around with the data, these are various filters that we use to exclude false positives. And this set of three, well, it's more traditional to throw away. So the major allele you call with the reference genome, the Portuguese one. So if there's an A in the Portuguese specimen and a T in some other ones, the A becomes the major, the T becomes the minor. And so here we're throwing away a little bit of the data is the way to think about it. Here we're throwing away more than half the data. And even if we throw away more than half the data, it doesn't take, and this is number of loci, so think about this as number of SNPs. And this is 100 SNPs. And I told you that the minimum difference between two closely related is 25,000 SNPs. So we'd have to throw away actually most of our data to call two mushrooms to be the same genetic individual. I hope that makes sense. But I'm happy to answer questions if it doesn't make sense. The message is basically, I think that it's okay. I think that our genomes are giving us accurate information. I think the AFLP actually gave us the same signal with lower resolution, which is that every mushroom is its own individual. This is an annual weedy fungus, so therefore it may be no surprise that it's invasive. It turns out that unbeknownst to us, because of a long story, actually with the original AFLP sampling, we also had some temporal data. And that temporal data had to do with this population that we call Césac that's growing with this single Lebanese cedar in France. And here again, here are AFLP data. So there's lower resolution, and we do find that some mushrooms belong to the same genets. If you see, I think I forgot to say, and I hope that it was clear that each one of these is a mushroom, and if it's on its own, then it's its own genetic individual. Here you see a cluster of, I think, 2 mushrooms that are called as the same genetic individual. But still, over time, even though you do find some genets that are made up of more than one mushroom, still the signal here, even with these old AFLP data, is that you have lots of individuals anytime that you sample. You don't find big mycelia. So I think, actually, maybe I'll just linger there and say that I think that story is complete. I think in my mind, and part of the reason I'm giving a talk is to get feedback on this, but in my mind, there's no controversy that the mechanism of a range expansion in this case is sexual spores. That's how it's getting around. And which implies, and also the data imply, that there are very small bodies. We're just not used to thinking, I don't think of ectomycorrhizal fungi that way, as being very weedy and having very small bodies and having a fast-paced reproductive output. But I think this life history is, I would suspect, is maybe not unusual, even though we haven't really thought about fungi that way before, necessarily, not ectomycorrhizal fungi. So this is just to show you, I guess it's just to show you that we can do this, which is we have now a better understanding of the population structure. So here you're looking at all the genomes and how they're related to each other. And you can see, for example, that the Serbian, so here's AFLP. You can see the Serbian population looks very different. This is basically the East Coast, and this is the West Coast, which is interesting. With the SNP data, though, with the genomes, there's something even more interesting, I think, which is check out these colors here. These are all from what we call Drake 3, 2004, 2014, and 2015. They're all here. In other words, when you sample through time, populations cluster not according to whether they were collected in 2004 or 2010. They cluster according to where you got them. So these guys are all from the same place. Across a decade, they still cluster together. These guys are all from the same place. Also, across a decade, they cluster together. So you cluster by space, not time. Which implies, because, oh, I hope the map's the next one. Yeah, so this is one of the populations. 90 meters across the road is the other population. For these guys to cluster by space over a decade and be distinct from these guys implies that, in contrast to what many people still think, spores do not travel far. And I would say that this is one line of evidence in a growing body of evidence that these kinds of spores do not travel. They do not make it up to the atmosphere and span the globe and settle again. It does not mean that all fungi are dispersal limited, just the same way that no one would ever get up here and tell you that all plants disperse this way or all animals disperse this way. I think it's time for us to move beyond a conversation where we tell each other that fungi do this or fungi do that. With 10 million species or 1 million or whatever it is, there is a range of life history strategies. And I think that's what I'm seeing here. But at least for phalloides, it's incredibly dispersal limited. So if you do a Mantel test, which is basically you correlate a matrix of geographic distances to a matrix of genetic distances, what you find is that if you look at the mushrooms from these two populations, genetic and geographic distances are strongly correlated. So that would be the formal proof, in a way, that these guys are really dispersal limited. So in the last 4 minutes, I'm going to tell you a very different kind of story that we're just starting on, just because it's really interesting, and I thought it would be fun to talk about it. So one of the things that I really resisted about working with the death cap is the fact that it kills you. And I think that's partly because everyone wants to talk to me about how you die when you eat the death cap. And the answer is really unpleasantly. You have vomiting and diarrhea, then it goes away, and then you forget that you ate a wild mushroom, and then about 16 hours later, you get violently ill again, and you go to a hospital, and you get put on palliative care, and either it works or it doesn't work. Sometimes you need a liver transplant, and it's really unpleasant. And my aunt, who really doesn't want to be in a nursing home, when she tells me, I know that I can come to you, I'm like, I would not kill you with the death cap because it's really just not the way to go. So that's a true story, by the way. So we do know something about the biochemistry, but only because these compounds have emerged as useful in medicine. So if you know anything about RNA polymerase and how it works, you know about RNA polymerase because of experiments that were done with amatoxins that actually very specifically stop RNA polymerase II from working. But there are other compounds that so for example, Sigma sometimes calls me and says, and you can't synthesize it, so they want to know if I can collect fresh material from them so that they can extract these things and sell them for $500 a gram or whatever. And they're really interesting, and you can tell they're really complex compounds, right? So they're amatoxins, they're phallotoxins. Phallotoxins are also useful in medicine sometimes, cycloamides. There are lots of these, and they're really complex molecules. They're actually, they're unusual because they're actually translated proteins. So a lot of times secondary metabolites in fungi don't go through the ribosomal machinery, but these guys do. Don't ask me how you put something that stops RNA polymerase II from working into your ribosome to synthesize it. That seems to be a question that actually nobody's really asked or answered. So I think these secondary metabolites are probably very expensive. And we know a little bit about them from our own kind of fooling around, that they're variable in nature. So I've started to really think hard about the question. So here you're looking at distributions in the cap and the stipe and the vulva at these same two sites, Drake 2 and Drake 3, and they're not the same. So they're genetically very different in their expression of these secondary metabolites. So one of the things that I've really started to think about with this fungus is how is it growing so abundantly in California? Is it an open niche that nothing has ever exploited before? So actually there is no natural selection, and it's just slipping in unawares? It's just like nothing else can do whatever it is that it's doing? Or is the story one about it's evolving in the habitat to to adapt. Actually, and I'm sorry, I forgot, I also want to just acknowledge, part of the reason that I wanted to do this is because my colleague Jonathan Walton, who is at Michigan State University, who has spent a career studying amatoxins, died about 2 weeks ago, or 1 week ago, actually. And so this is a little bit also an homage to Jonathan Walton. I think he would love these data that I never got a chance to show him. So I am thinking about what adaptation would look like in this fungus, and it just struck me that this thing that I haven't wanted to talk about for so long maybe might be a really interesting thing to study, because we think about enemy release for invasive species, and we think that these might be defensive compounds. So if this has really reached California and escaped its enemies, then maybe it doesn't need these defensive compounds anymore. Anyway, it seems like an interesting thing to ask, which I don't know why it never occurred to me before. So Sam Harrow has taken these genome data, and she's started to ask the question with these 86 genomes, what do the genes look like? And actually, we have a really, because of Jonathan Walton, we have a really good understanding of what the genetic basis of all these genes is. And so we're asking about all the different kinds of differentness that we might find among the genes. So maybe we find the same gene in a different location, or a different copy number, or the same gene in the same location but a different allele of that gene, or maybe we find a different gene in the same location. So there seems to be all kinds of interesting possibilities of things that we could ask. And actually, it seems we have now analyzed 3 genomes, and it does seem to be a really interesting answer. And this is very preliminary. The blue are the 2 California genomes that we've compared to each other, and they have lots, so I'm not going to go into the details of the different kinds of differentness too much, except this is basically, these are homologous genes, so they're in the same position, but they're different alleles. So it's in this example, I'm going to show you that kind of differentness. So these 2 California ones are more different than either is to the Portuguese one that we have examined, which certainly at least is suggestive, and there's a lot more work to do, that there's a lot of dynamic evolution associated with these. So here, just to dig into the data a little bit more, you're looking at amino acid sequences. So here, the 2 California ones have different types of alpha ammonidin. It's a flavor of ammonidin. But the California one looks like the Portuguese one. Various flavors of the beta ammonidin. Anyway, you can see how it goes. What's particularly interesting are these cycloamides, which nobody knows what they do. And it turns out that they are either present or absent in different ways for the 3 different specimens. So these guys always seem to be there, but these guys are not. So they seem much more variable whether you have them or you don't. But we know nothing about the ecology of why these are toxic. They're not toxic to kill humans. That's not the point. So what is the point? I don't know. No one's ever done any work to explore that. So if anyone has ideas, if you can look at these structures, and if those structures speak to you in terms of what they may do, I'd be really glad to, I don't know, to chat about it. It'd be useful. Okay, so, all right, so I do have 2 more minutes. So that's the end of my formal talk. But I want to reiterate kind of how I started the talk, which is that we know very little about fungal biodiversity, so little that it's painful. And so here's just maybe a fun story to leave you with that thought, which is where I started and where I really want to end. So Amanita inopinata is the unexpected Amanita. And it was first described by Reed in 1987 from the UK, and it was described as an introduction. So Reed said, this is not a fungus that's native to the UK, but I'm finding it here. And this is very common with Amanita, for example, that they're described for the first time from an introduced habitat, not a native habitat, which is just so, I don't even know what to say about that, but that's true. So it was first described then. And then Jeffrey Kibbe, another guy, published in this journal, Field Mycology, a fungal portrait. And there you're looking at Amanita inopinata. And interestingly, he said, oh, and Jeffrey Kibbe, by the way, lives in New Zealand. So he publishes this thing called, a fungal portrait. And in it he says, as to where it comes from, I do not consider it native to New Zealand and speculate that it comes from Europe or North America. This engendered a swift retort by Reed, by Kees Baas, who said, yes, I said Amanita, and here you can see Kees Baas actually entering the story, is watching Amanita inopinata move from the UK to the Netherlands. Thus the title, The Unexpected One Jumped the North Sea. Yes, A. inopinata, but that species is known only from New Zealand and southeastern England, are the spores amyloid. So he wants to know what it really is going on. My heart really made a little jump when I immediately recognized it as A. inopinata, not least because of the color photographs published this year in Field Ecology. This was really an exciting discovery, and I nearly kissed the finder, but instead we danced a bit around the table. So he's happy to see the invasion front expanding to the Netherlands. But the point of the story is that the people in New Zealand think it comes from Europe, but the people in Europe are speculating that actually it may have come from New Zealand. And the truth is that we have no idea what the native range is of this fungus. And this is not the only story I could tell you like this, where we really don't know. We really don't know what the native range is of the fungi that we're describing from their introduced ranges. So it just seems really odd. So the world is changing, and the biogeographies of fungi must also be changing. But because we have such a poor understanding of native ranges and baseline biodiversity, we don't really understand what's going on. Although, of course, some of this record may be preserved usefully in collections. And so I want to leave you with these questions of how, and does it matter? And I published a paper called Last Chance to Know. And that title, Last Chance to Know, comes very directly from this book by Douglas Adams, who published The Hitchhiker's Guide to the Galaxy, where he went around the world and saw things that were going extinct. And he called his book, and it's fantastic if you haven't read it, Last Chance to See. And I would argue that for us, we're not even quite there yet. We're still just at the last chance to know. And with that, I'll thank my funding sources. And if you want to see me talk more, I have 3 iBiology talks up, and they're actually really great for teaching. If you're ever teaching intro bio or a field course, and you need to, sometimes I'm asked about where to go to learn about fungi, and so I actually just did something so that you can learn about fungi, and it's on iBiology. And I'll thank my lab. Denny and Jacob are here. Sam, who's doing the toxin work, is here. And with that, I'll be happy to take any questions. Thank you. Yeah.
Given that it's the Amanita phalloides is so dispersal limited, do you think that the reason why it spread all along the Pacific coast is because humans have been moving it to all those different
I kind of think so, yeah. And I think very specifically, people do like to plant Quercus agrifolia. So I think the story of its long distance movement is a story of moving trees with soil. And I think, it had never occurred to me until I started doing this work that, of course, every time you put a plant into your garden, you're not just putting that plant, but you're putting everything that's in the soil around that plant as well. So, yeah, I think that's what's going on. Yeah. Yeah?
Given your remarks of the last few minutes, is there any way you can rule out the possibility that actually this Amanita phalloides is actually native?
Yeah, no, that's a completely fair question. And actually, that was why it took so long to build this story up, is because when I first started talking about it, that was the question I got more often than anything else was, are you sure that it's not native to California? And so, so it's so in fact, the paper Last Chance to Know is really an existential riff on what we can know and what we cannot know. But what I did is I went back to all the specimens that had been collected and labeled as phalloides, and I did ancient DNA work to figure out that all of them were wrong up until a certain date. And actually, if you do and then I did at the time Sanger sequencing of individual loci to show that actually also phylogenetically, it's not its own isolated endemic population, but it actually is related to various populations in Europe in a way that can only be explained if it were introduced and was invasive. But that's exactly, and it's actually, what I think is so interesting about that question is when I look at all the work that had to be done to establish the baseline data for the death cap, in a way, I'm not surprised that we don't have more evidence for it. If you want to know if a plant is invasive, you can look it up in a USDA database. It tells you the native range. It tells you where it's been introduced. If you pick up a field guide to mushrooms, most of them do not have range maps in them. So yeah, no, I think it's a very complicated. And in fact, when we first started looking at the genome data, there was something about it, the Portuguese populations, where I was like, what is going on here? And then it took me a while to understand that actually what I was looking at was probably a signal of multiple introductions from Europe to California, and that explained that data. But I had a heart-stopping moment. I have to say that now I would answer that question differently in the sense that I don't think you see the kinds of patterns that you see in terms of abundance, spore carp production, et cetera. Those are not the... Those are also signs to me. It behaves so differently in California than in Europe, and in all the ways one might expect of an invasive species. That, to me, the ecological data becomes more convincing to me than anything else, actually. Yeah. 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 I think it's a parasite. I think the root tip, I think it has, so if you look below ground, the root tips don't dominate. So Phalloides root tips, you basically have to dig right underneath the mushroom. If you dig right underneath the mushroom, you find Phalloides root tips, which in retrospect, given these data, makes a lot of sense. If you move even a little bit away, you don't find that. So if I had to guess, I would say, and I would love to figure out how to prove this, that this is not an ectomycorrhizal mutualist, that this, the spores land, they germinate, they mate, they grow into a mycelium very quickly. They have a few root tips. They suck the carbon out of the plant. They give nothing in return. They make an enormous fruit body with lots of spores. They move on. That's what I think is going on. But it's really hard to prove that. Yeah?
I was intrigued by your conclusion that spores don't travel far.
Yeah.
So I wonder, in 10 years period, and that's not a lot of time for the plant partners, the pines or the oaks, whatever others.
Yeah.
Grow. So I wonder if you check the plant partner or host range.
The host range, like what it grows with in terms of the host?
The mushroom.
Yeah, we know its host range very well, actually. I didn't talk about it, but we published a paper on this where we show its host range. Am I understanding your question well? What plants does it associate with?
Yeah.
Okay, so in Europe, it's mostly, it's oaks or pines, depending on where you are. On the East Coast, pines, only pines. On the West Coast, oaks, but one oak, Quercus agrifolia. In California, more than 90% of the collections are associating with coast live oak only. So, and we know that not just from herbarium specimens, although that's an important source of data, but also because we collected root tips, which are the site of the symbiosis, and we sequenced the fungus and the plant from that root tip. So we know very precisely for those root tips in particular what the host associations are.
But what makes me think is that in 10 years, the plant may have not changed its range much, so then your fungus cannot change.
Right, so right now it's still constrained within the range of Quercus agrifolia, but I would predict actually that it will expand to fill, well, as long as the abiotic parameters are suitable, that it would expand to fill the range of Quercus agrifolia. So it's still an active invasion. It's not at equilibrium. I haven't done that work in a while. I mean, I think, of course, another question I think that you're asking is, if I waited another 10 years, would I see a different pattern? And as to that, I can say, obviously, I don't really know. I haven't done that. But I think, I feel the fact that I'm not seeing any mushrooms come up with multiple genets in 2004, 2014, 2015, I think the simplest explanation for that is that they really are small body sizes, small genets. And the patterns, yeah, so maybe I think I'm answering a lot more than you asked, but I think the data certainly suggests that the spores don't get very far. Yeah.
Because in your talk earlier, when you say that when you had a pine, but not mushroom, the pine didn't grow or germinate. So what I'm thinking is that if the plant didn't get there, and if you have that spore, they will not germinate.
Yeah, but, sorry. And so what I should have said is that in these communities, Phalloides is not the only ectomycorrhizal fungus. So the plant is interacting with hundreds of other fungi simultaneously. So although I told a story just about the death cap, the plant and Quercus agrifolia elsewhere would have lots of other partners that it was interacting with. Yeah.
You said that you know several genes which are responsible for production of these toxins.
Yeah.
I wonder what is known about phylogenetics of the genes. Are these toxins evolutionarily young? Can we see signatures of positive selection, what is the origin of
No one has ever asked that question.
But it's pretty straightforward.
It should be pretty straightforward. I can tell you that if you do a phylogeny of the genus Amanita, not all Amanita are deadly poisonous. It's just
Edible.
some are edible, exactly. So there's a small cluster, actually not so small, but there's a cluster of toxic Amanitas. So clearly the origin of the toxin genes lies somewhere in the history of that particular group. But to my knowledge, no one has ever done a population genetic study of the toxin genes.
But did you blast these genes against the database?
To see whether there are matches?
Yeah, to see what they originated from.
Maybe I don't understand. We haven't, I mean, we know what they are because people have characterized them really thoroughly in biomedical contexts. So I don't feel like we need to blast them. You're wondering whether I would get a hit to some other fungus?
These genes probably evolved to synthesize these toxins pretty recently. And likely they're not de novo genes, they originated from something.
No, I don't. I think whatever. I think I understand your question. I think we've lost that signal. I don't think that. I think they're dynamic enough that there isn't anything to relate them to, before the Amanita. Although it's an interesting question. There might be clever ways to do that if we had some genomes of closely related non-toxic species, for example. Yeah, but I don't think we have those genomes. But it would be a really interesting thing to propose to do, get one of those genomes and see kind of where they are. We're definitely thinking about the physical location of them. So I have a colleague, Nancy Keller, who works with secondary metabolites and ascomycetes, not basidiomycetes, and she's convinced that these guys are all going to be physically all next to each other physically adjacent to each other in a cluster. Yeah, I hope so. And I think we have a really interesting ecological context in which to do that because we have this invasive range and a range where it's not and a range where it's native. So I think it has, but of course the thing that I can't answer for you, even if I found they were under selection, I have no idea selection for what, because we have no idea ecologically what they're doing. They kill dogs, they kill deer. We think they kill deer. I know they kill dogs because, again, I get a lot of calls about that. But I don't think that they exist to kill dogs or deer. So what they do, I don't think they're antivirals. They don't look like antivirals, according to people I know who would know that better than I would. We don't know what the variation would be. Like if we have 2 alpha aminidins, is that variation neutral? Does it make a peptide that's different in some relevant way? I just amazing. Actually, the more we kind of move into that world, the more I think there's a lot of interesting.
I think these days to sequence genomes of 10 different amino acids should be
trivial. Yeah, should be trivial. Yeah. The only thing that sometimes gets in the way is finding the specimens themselves. But yeah, no, I completely agree with you. And it would be interesting to go up and down the phylogeny. Oh, another interesting thing is we know they've been horizontally transferred to a different genus of fungi, the Galerina, which also have amatoxins. And HGT of secondary metabolite clusters also appears to be a theme that's emerging in the study of fungi.
Okay. Well,
Yeah.
let's thank Anne.
Thank you.